Hi everyone, my name is Erin and I'm one of the Ensembl outreach officers. Welcome to today's video on comparative genomics data in Ensembl. This is part 2, where we're going to be looking at sequence-based resources. So this is predominantly multiple and pairwise genome alignments, which are used to infer things such as Synteny and ancestral sequences and so on. So today we're going to start with a short presentation and then go straight into the demo. And if you're only interested in the demo, feel free to skip ahead to the time shown below. Otherwise, let's get started. Okay, so as I said previously, this is part 2. The first part of this video series was on gene-based resources, so looking at phylogenetic trees and how we infer homology, including orthology and pairology, stable ID mapping and protein families, and you can find this video on our YouTube channel. And today we're going to be looking at sequence-based resources, which are found in the location tab. So these are whole genome alignments, both pairwise and multiple. And these are used to infer ancestral sequences, age of base, conservation scores and constrained elements, and Syntenes, which we're going to go to in a bit of detail now. So we have two types of alignments, both pairwise and multiple alignments. These are derived from different alignment methods. The pairwise ones we use last Z, whereas the multiple alignments use a range of different methods, which I'll talk about briefly for different groups, depending on their evolutionary distance. Pairwise alignments are obviously pairwise, so between two species, whereas multiple alignments can be between many species, and like I said, depending on the method, we have groups between 4 and 70 in species number. The pairwise alignments require chromosome level assemblies, so typically quite high quality and high depth assemblies. Multiple alignments can use either chromosome level assemblies or scaffold level assemblies, where we have the whole genome sequence but it's not yet been assembled into chromosomes. So pairwise alignments are used for identifying regions of Synteny, and we'll talk about this in the next few slides, and also useful for scoring ortholog quality, which is what we looked at in the previous presentation. Multiple alignments, on the other hand, are used for identifying ancestral sequences, the age of base, conservation scores, and constrained elements, so really important for identifying evolutionarily conserved regions. So pairwise alignments can be found on both the Ensembl main site for vertebrates, ensembl.org, and also our sister site Ensembl Genomes, which covers other taxonomic groups, such as plants and protists and metazoa. Multiple alignments, however, are only for the Ensembl vertebrate site. This is because it's a smaller evolutionary distance within the vertebrates compared to really larger groups represented on the Ensembl genome sites. So let's take a look at whole genome alignments now. We're looking at pairwise alignments here. So these are aligned, as I said before, with either BLAST-Z, which is a slightly older method, and LAST-Z, which is the newer method that we're currently using. You'll see these referred to throughout Ensembl, which is why I mentioned them here. For some species in Ensembl genomes, particularly in the plant species, we're using this translated black net as a different method, so you may see that referred to there. You can see a full list of species with pairwise alignments and the methods used to generate these alignments at these two links here, which we'll have a look at in the demo. So we measure Synteny based on data from pairwise alignments, which we calculate by looking for alignment blocks. These blocks contain genes that are in the same order in both of the genomes, and when these blocks are less than 200 kilobases away from each other, we group these again into a larger block. If these groups have less than 2 non-syntenic groups, so regions where the gene order is not conserved, separating these two larger blocks, and if they're less than 3 megabases apart, then we link these groups as syntenic blocks. So these can be very large regions of the chromosome that are mapped to different species. It doesn't necessarily mean that within that mega block that the gene order is consistent throughout, but it's predominantly consistent between the two species. So multiple alignments, as I said, we have a number of different methods. They all stem mostly from this PCAN algorithm. So the first two, Enredo, PCAN, Orpheus, we have both a high coverage and low coverage version of this. So the high coverage one is, as you can see, between quite small groups, particularly closely related ones, so sauropsids, so predominantly lizards and birds, fish, primates, and eutherian mammals. So the EPO low coverage encapsulates groups slightly larger and slightly more diverse, but also ones with slightly less coverage genome assemblies, so including the scaffold level assemblies I mentioned earlier. So essentially this is just a less stringent pipeline, which allows for these differences in taxonomy and also sequence coverage. The Mercator PCAN analysis is again another type of pipeline that incorporates both mammals and birds, so a higher quality assemblies at the chromosome level, but for a slightly larger taxonomically diverse group. And the Cactus Pipeline is a newer pipeline that we've used to align our Murinae. So this is a subfamily of the Muridae, which includes mouse, Algerian mouse, and all of the mouse strains, as well as rat. Okay, that sums up the very brief presentation on introducing the different types of alignment we have and the alignment methods. You can read more information about this, which goes into more detail about the alignment methods and pipelines in our Ensembl Comparative Genomics resource paper. So I'm just going to jump out and we're going to go to ensembl.org to go to the demo. Okay, so I've come to ensembl.org, the main vertebrates site. As you can see, we have this blue bar at the top, which is the header. This contains quick links to tools such as the VEP and Biomart. And on the right-hand side, you can see that we've got the quick search box. The main page contains the main search box, a list of all your favorite genomes and all of the genomes in Ensembl. On the right-hand side, we have release news. So currently we're on release 93. It's important to make a note of which release you're using, as we update our databases every 3 to 4 months. So you may find that some of the data has changed, and if you make a note of the release you're using, you'll be able to revisit this in our archive sites and keep track of what data has changed. So in the previous video on gene-based resources, we took a look at the gene TP53, and we're going to stick with this for looking at sequence-based resources today as well. So I'm going to come down here and type in TP53 into the search box and click enter. So we saw this page in the last presentation. And I'm going to click quickly on the link down here, which says to go to location, which is going to take us to the location page for this gene. So if I click on this, instead of going to the gene tab at the top here, we've come directly to the location page. And automatically we're put onto the region in detail page as well. We have a section called Comparative Genomics, which is broken down into Synteny, alignments as an image, alignments as text, and region comparison. I'm going to click on alignments text to explore this first. Okay, so we come to this page where you can see our location at the top, and we're highlighted by this red line on the first image here. So this is our location on chromosome 17. We have a help button question mark here, which you can click on, and that will open help specific to this page in a new tab. And you can see that we have no data on this page at the moment because we have yet to select an alignment. Let's click on select alignments here. So this opens a pop-up. At the top of the page, you can see a prompt to start typing in the common or scientific name of a species. And there are also several buttons to browse species by taxonomic group split into multiple and pairwise. On the right, we have a panel where our selected species will appear. Currently, this is empty, as you can see. So I'm going to start by clicking on multiple. And you can see we're presented with a range of different options. Depending on what species you're working in, these options may be different. So these are labelled by taxonomic group and also the alignment methods that are used to generate these alignments, which I mentioned in the presentation. So this list will contain different alignments depending on what species you're working on. We're currently looking at gene and human is aligned to most of the species in Ensembl, and humans are primates. So we have a small set of primate species which have high coverage alignments. If you're working in mouse, for example, you'd see the Murinae group here instead. You can click the checkbox to choose all of the species in this group, or you can click on this plus to expand and just choose select species from a certain group. So if I click to turn this off, I can choose chimpanzee, gorilla, and orangutan, for example. And you can see these have appeared in the right-hand side panel, and the count box here is showing me how many we have. You can also search for species, as I mentioned, so let's type in dog. And it comes up with a suggestion based on what you've typed in, so you can search for either the common name dog or the Latin scientific name, Canis familiaris. If I click on this, you'll notice that my selected species is only reduced down to dog. This is because dog is not within the 12 primate EPO alignment group that we previously selected, but instead we're presented with a pairwise alignment. We can choose to view both primates and dog alignments, but they need to be aligned with the same method, which is why the others disappeared. If we click back to all alignments at the top here, click on multiple and expand this section on 26 eutherian mammals, you can see that we have dog within this group, along with our chimpanzee and also our orangutan. So let's choose those three: chimpanzee, dog, and orangutan. I don't need to select human because we've come from that page, so it's automatically going to do that alignment anyway. You can click apply. Okay, so you can see that the page is reloaded. We're presented with the option to download the alignment, which is grayed out currently as we have 2 alignment blocks present, which I'll come back to in just a moment. We can also see that we have this hidden box, which shows us the 23 species in the alignment that we are choosing not to show because I didn't select them in the pop-up. Okay, we come to a table with the 2 alignment blocks that have been found for this region. You may find that you have more or fewer blocks present, depending on what alignment you're looking at and what region you're in as well. So we have 2 blocks here of similar size. You can see that they're both about 13,000 base pairs in length. And if you look at the location on human, you can tell that these come from 2 neighbouring regions. The first block has alignment with 23 species in this group, plus the 3 that we're already looking at, so the whole group of species in the 26 eutherian mammals have alignment with this section of the genome. The second block, block 2, only has alignment with 12 species, and we're going to explore this further in this case. So I'm going to click on this link here to view block 2. Okay, so we come to a similar page, and you can see that we have the download alignment button, which again we're going to come back to show you the different options, but essentially this isn't grayed out anymore because we've chosen one alignment block. Because it has these certain species in it, it didn't correspond to the other block, which aligned to a larger number of species. So you need to choose a block before you're able to download the text alignment. And if we scroll down, we've got some new information compared to the previous page. So first we're presented with this taxonomic tree image with alignments shown in blue. This looks similar to the gene tree image that we saw in the gene page from the previous presentation, but it is slightly different. In the gene tree, we had different types of nodes for speciation and duplication, and we were able to view paralogs of genes from across the genome. This image instead is only showing speciation and no duplication nodes, as we're only looking at the DNA alignments at a single region. The blue alignments on the left showed DNA alignments and the gaps shown in white. In the gene tree, the alignments were shown in green, and these represented alignments of the protein sequence of that gene. Now we're looking at whole genome alignments, so looking at the whole DNA within this region rather than just the protein translation. We can see that there are some differences in alignments between the species. Human and chimp are fairly similar, and orangutan again is more similar than dog, which is a more distantly related species. So below this image we have links to the aligned regions at the location in the different species. These IDs are split up into chromosome at the beginning, followed by the assembly version, followed by the chromosome number, start coordinate, end coordinate, and strand. We can see that this region is on chromosome 17 for all of the primates, including human, but on chromosome 5 for dog. And we can see that all the regions align on the forward strand as indicated by the 1 at the end, and if it was on the negative strand it would be marked with a -1 to indicate the reverse or negative strand. Finally, we have what we're interested in, the actual sequence alignments. I'm going to click on display full alignment, as it by default only shows the first 120 columns. So here we see our species names, human, chimpanzee, orangutan, and dog, all aligned together. The red text is showing exons of the gene within that species at the aligned region, so you can see that they don't always necessarily match across different species. The black text is either intronic or intergenic sequence, and hyphens represent where a species has no alignment at this location. Remember that we're excluding a number of species from this alignment group, so we can assume in regions such as this, where there are all 4 species with no alignment at this region, that another species in the group that we're looking at, this 26 eutherian mammals, has alignment at this location. We can check this by scrolling up and choosing to view all the species within this group. So if I click here and select another alignment, we've got our 3 species selected here. I can just click and add all of them, so we now have our 26 eutherian mammals. If I click apply, the page reloads and we'll be able to see the alignment for all of these. So again, we're presented with a larger tree showing all of the species, which we know to be 12 from the information we saw in the block, aligned at this location. And again, I'm going to click display full alignment. Okay, and in this example here, you can see that most of the species don't have any alignment here, but the horse alignment shows an insertion of sequence between regions of alignment with other species. We can add some additional useful information to this alignment. If we scroll up, we find the configure this page option on the left. There are many options here which enable you to view variants and alter the formatting, which I'm not going to change for now, but you might find some of these useful. I'm going to first of all change the codons from don't show codons to start stop codons. I'm also going to tick the show conservation regions block. And I'm going to click the tick and close the box. Once again, I need to click display full alignment. Here we can see that the background of much of the sequence to begin with has been coloured blue to highlight the conservation regions, which is where the majority of the species in this group have alignments. If we scroll down, we can start to see the yellow blocks, which indicate the start and stop codons. Once you have the species of interest and the particular alignment blocks, you might want to export the sequence. If we scroll up, we can find the download alignment option. Here we have a pop-up now with many multiple examples of the types of file formats. The faster format is automatically selected. However, please note that this was going to download the data in blocks. So if we click to view preview, what we'll get is the alignment for human, followed by the alignment for orangutan, for example, and then the other species that you've chosen. So this may not be particularly useful if you're looking to compare the species directly. If you've added useful information through the Configure This page, you can also download this annotation with the markup. If we click on RTF, rich text format, you can see this example has highlighted different colours indicating variants, for example. And again, we're presented with this page, which is similar to the one we found in the Configure This page, where you can add and remove different formatting options and different attributes for the sequence. Many of the other formats that you can download, such as ClustalW and Mega, are more commonly used for alignments and may be useful in downstream processing or creating trees of your own, for example. You can preview all of these and how your data looks in it for every one apart from the RTF format. Okay, I'm going to click to close this now. While this view is interesting and you can highlight exons in different species, you may find it easier if you're comparing gene models within a region to use the alignments image. Okay, so now we're on the alignments image page. As we already selected block 2 on the last page and all species in the 26 eutherian mammals, then this is what is shown on the alignments image page by default. If you come to this page first, then you will need to choose an alignment block and also your alignment to begin with. So first of all, let's change our species selection to our 4 species from earlier: human, chimpanzee, orangutan, and dog. This makes the comparison much easier. So if I scroll down below the first 2 images, I can click to select another alignment. I'm going to untick this, click chimpanzee, dog, and orangutan, and apply. So we can see the model of TP53 shown. This view collapses them into a single gene model rather than showing all of the different transcripts, but you can change this back if you're interested in looking across all transcripts. We can see that there are some differences in the start positions, and this is a negatively stranded gene, so we're looking at transcription starting over here. So this is exon 1. You can see that for chimpanzee, it's missing this exon compared to the other species. The aligned sections are highlighted with this red background colour, and the legend shows how an event such as an inversion or an indel will be highlighted on the display with these small triangles. And if we zoom out, we may be able to see some extra information here. Yeah, so we can see here that we have these triangles, which if we scroll down, we can see represent indels. While this display may be useful, particularly for comparing larger groups and multiple alignments, it's really just displaying exactly what was shown in the alignments text image, but in a more visual interpretable format. What may be more useful is using the region comparison page. So let's view that now. The region comparison page offers a little more context because we can look at an overall region rather than just a specific block. It also offers alignments at the different levels, starting at the chromosome level, progressing to an image at 1 megabase or 1 million base pairs at the region of interest you selected. This is different to the previous page because the alignments are generated only from the pairwise alignments, rather than the multiple species alignments that we were looking at previously. However, you can still select multiple species from different taxonomic groups, so we can select our chimp, orangutan, and dog again. So let's do just that. So you can see here we're not presented with the option of multiple versus pairwise because it's pairwise by default. But what we can do is click on primates, find our chimpanzee and orangutan. And if I click on other mammals and carnivores, we can find our dog here as well. Don't forget that you can always just search at the top here. So again, compared to the previous page, the alignment image page, we have the alignments represented at all of these different levels, starting with the chromosome and where these are located on that chromosome. So again, we can see that Canis familiaris, CFAM, represented here, dog, has alignment on chromosome 5 compared to the others on chromosome 17. The middle image allows you to see whether the genes in this region are similar between the different species, so this may be useful for you if you're comparing a large region, for example. But at the bottom is the most detailed image and usually the most useful. This image shows you the alignment blocks and also the links between them, which is useful for checking for rearrangements and translocations. Sometimes there are gaps in the genome sequence itself for particular species, which you can see represented by a white space in this blue bar that represents the contigs. Because these images are generated by pairwise alignments with human, the human region will be displayed more than once to neighbour the other species' genomes. So you can see highlighted in pink, we have human represented both here and here to enable easy comparison between dog and human and orangutan, and human and chimpanzee. Similar to the region and detail page, you are able to add on extra information to these images through the Configure This Page option, so you could add variants and so on. It's important to note that not all species in Ensembl have the same data available, so you'll need to configure information for each species independently. So let's have a look at how to do that now. If we click on Configure This Page, I scroll back up. You can see here that we come to a Configure This Page pop-up, which is similar to the region and detail page if you're familiar with that. We have at the option this additional option to choose which species to configure. So by default it's human, you can choose from the species you have selected. And also you can save your own configuration. So you can use the default, and this shows you the active tracks that are present in the default. And then whenever you add extra tracks, you have the option to save it if you're going to do repeated analysis, for example. You can configure the way that the comparative features are displayed. So if, for example, you are looking at a gene-dense region, you might find it useful to link up the genes between the different species. So if we scroll down here, you can see you have comparative features as an option, and an option to join genes, as well as changing the alignments formats from compact to normal. I'm not going to change these, I'll leave these as default. You may also be interested in comparing gene models from a different database, such as RefSeq, to see if they have different gene predictions to the Ensembl gene models. And you can add these to the display also. So if I go to genes and transcripts, for example, and scroll down, I can select RefSeq annotation import. I'm going to choose collapsed with labels, so I only get one gene result rather than all the different transcripts. If I scroll up, I can change different species called dog, and I can go to genes and transcripts, add in my RefSeq collapsed with labels. As you can see, we have far fewer options for dog than we do for human, which is understandable as human is much widely used. Orangutan does not have RefSeq alignment, so we can't add these on for orangutan. For chimpanzee, we do have these data, so I click on genes and transcripts. Again, I can add this collapsed with labels. If you find any other interesting features that you'd like to add on, you can always click on the eye if you're not sure of what they mean, which usually have useful definitions and links, so you feel free to browse these for finding other additional interesting information. I'm going to click and save close. And if we scroll back down to our more detailed image, we can see that we have new gene models highlighted. If you click on them, you can see where the analysis has come from. And over here, you can click on and see the different types of data tracks. It pops up and tells you what it is, and quick links to change the display option as well if you're interested in, and also quickly turn off the tracker if you want. So what we can see here is that the RefSeq annotation import for some of the species is quite different compared to the Ensembl one. So it's always useful to compare across different annotation methods. And again, we have these blue lines, which are showing us the links between the genes. And note this is only doing it for the Ensembl gene models and not the RIF-seq gene models, but it does enable you to quickly find linked up species between human and the other pairwise alignments. So I've mentioned the region in detail page, and if you're wanting to view alignments in a single image, you can do this from this page, which is where you can also add information such as age of base, conservation, and constrained element scores. So we're going to have a look at that now. So we have other videos available for using the region in detail page, so I'm not going to cover it in too much detail here. But essentially, it's very similar to the region comparison page that we looked at previously, in that it's separated into 3 images which progress in complexity and zoom into the region of interest. So the region that we're interested in is highlighted with the red box in the top 2 images, and we can add a huge amount to these displays from the Configure This Page panel. So let's click on that to explore what comparative genomics data we have available. We're shown here all of the active tracks, and this looks very similar to the Configure This Page from the region comparison, but with some more options available. So if we scroll down, we can see now that we have this comparative genomics section. This section is split into 3, so first of all, multiple alignments, conservation regions, and lastly alignments, which are actually just all the pairwise alignments we have available for the species of interest. You can see some options such as constrained elements for 70 eutherian mammals and age of base are turned on by default. And I'm going to turn on a few other options here to explore. So I'm going to turn on a multiple alignment for the 32 amniotic vertebrates. I'm going to turn off this 70 eutherian mammals and instead turn on the conservation score for 32 amniotic vertebrates and constrained elements as well. So the conservation scores, if you're not sure of anything, we have these little eyes here in the far side that you can turn on, which give you sometimes links to more useful information. So these are derived from this pipeline called GERP conservation scores. So these are calculated on a base-to-base basis. So each nucleotide is examined independently, and it indicates how many species from the alignments have an exact match at this locus. The constrained elements are actually sections of the alignment which are slightly larger, which are based on these single base conservation scores, and they indicate which ones of these are highly conserved over a larger region. And this GERP test enables us to find these regions of a genome that are more conserved than expected by random chance. I'm also going to turn on some pairwise alignments. So for the species we were looking at earlier, so chimpanzee, I'm going to turn on compact. I'm going to turn dog back on as well, and also orangutan. Okay. And while I'm here, I know that in Ensembl we by default show all of the transcripts of a gene, which is very important to consider. But for the purposes of this demonstration, I'm just going to collapse them into a single model to make it easier to look at the whole image. So you can see we have all these transcripts that are going to get condensed, and our new data is appearing as well. We've got CCDS as well, which is showing all of the consensus protein coding sequences, which I'm going to turn off because it's quite large. Okay, so at the top of the image you can see that we have these 32-way GERP elements and 32-way GERP scores. So the scores are the conservation elements, so looking at each individual base and seeing how many of the species within this group have matches to human at this base. The constrained elements are shown as blocks indicating the conserved regions, which you can see match these peaks. Below this we can see the 32 amniotic vertebrates, represented by a pink bar. We have good alignment at this region across all of the different species. There's no gap suggesting that at least one species from this group has alignment in this region. Other features such as age of base and the independent pairwise alignments are shown at the bottom of the display, but remember that you can use these double-headed arrows to click and drag and bring things closer together. So I'm going to move these all together so we can compare. You can see that the age of base alignment has too many features to show on the scale, so we may as well zoom in to look at a region of interest. So at the top we have these two options, drag and select. If you choose this, it means you can just click and drag and move forward and reverse through the genome. This dotted line means that you can select a region, so actually what we can do is highlight this region of interest, And we're presented with an option to mark the region, which means that we can find it at a later date. And also to, if I remove this, we can jump to the region, which is zooming is in a specific area. You can see here now that the age of base is populated with these different colours and blocks. So if you scroll down, you'll see in the legend, we have red representing a human-specific base, blue is appearing in primates, and grey is just appearing in mammals, so different levels to examine. And again, you can see these peaks on the scores matching up with these constrained elements. We have gaps in the alignments here, and you can see that typically this is a very low conservation score area, which indicates that this is not a particularly highly conserved region. If you find an interesting region in one of the species you're looking at, you can always click on it and jump to view that region in that species, or look at some of these other pages that we've looked at earlier, so the alignments, text, image, or region comparison view. If you've identified a really interesting region, again, you can always share this with your colleagues. So if you click on this, you'll get this stable URL, which enables you to just send it to a colleague and it will remember the region you're looking at and all of the different tracks you've got turned on, and even if you've moved them around as well, like I have. And if you want to download the image, you can do so clicking here, which we saw previously in the last presentation. So a number of different formats, you can always see what they represent here. So now we've looked at these region images quite a lot, so let's have a look at Synteny now. And I'm going to click back into the Comparative Genomics section and look at Synteny. So Synteny is the conserved order of genomic blocks between species, or essentially regions of the chromosome of two species that have the same gene order. As we covered in the presentation, this is calculated based on pairwise alignment data, so we can only ever compare between two species at this time that have this data available. So by default, we're looking at mouse. I'm going to change this to look at chimp. And click go. So we can see human chromosome 17 shown in the centre and two chimpanzee chromosomes on either side. The colours are used to show the location on the second species, in this case chimp. So the pink is representing all of the alignments between human and chimpanzee chromosome 17, and the green, which is a very small region, is showing the alignments between human chromosome 17 and chimp chromosome 1. You can see that the majority aligns to the chimpanzee chromosome 17, but you can see that from the lines that the order of these segments is not necessarily conserved. So red lines indicate regions with the opposite orientation, while the black lines link the blocks, indicating that they have the same orientation both on human and in chimp. So both at the very top and at the very bottom, these are conserved, but these regions in the middle are generally a bit more mixed up. You may have noticed this red box on the human image, which tells us what region we're looking at. And if we scroll down, we can see a summary of this region and of all the genes that are contained there and where that maps to on the second species. So initially we have Homo sapien genes, so TP53 represented here, its location on the human chromosome, and then pan-troglodytes, which is the chimp. We can see that we've got TP53 again, so it's the same gene, and it tells us exactly where it's located, and a quick option to view our region comparison page if we want to look at that in more detail. Let's now have a look at dog, see how this looks. Click go. You can see that the split is a bit different compared to chimp. It's a more distantly related species, so we can see that syntenic regions of chromosome 17 are actually located on chromosome 9 and 5 in dog. And again, if we look at even more distantly related species, such as zebrafish, for example, much more distantly related to human, and you can see that the syntenic blocks here are much smaller than they were for both chimp and dog, which are mammals. Okay, so I think that's comprehensively covered the different sequence-based comparative genomics that we have in Ensembl from the location tab. If you want to find out more details about comparative genomics data in Ensembl, you can visit our help and documentation and visit the comparative genomics section, which you can find from the toolbar in help and documentation. We have annotation prediction here, so we go to comparative genomics. You can click here to see different types of alignments. So we have the genome analyses, which is what we looked at today, compared to the gene analysis, which is what was covered in the previous video. We can click on pairwise alignments. And you can see all of the different species here and how many different alignments they have. So for human, our most popular species, obviously, we have 98 alignments and 36 Syntenys, whereas for other less well-studied species, we have fewer alignments available. If you want to have a look at this for Ensembl genomes, if we click and go to ensemblgenomes.org, we come to the slightly different page, and we can go to data types. And if we scroll down, we have a section here on Comparative Genomics. Click on Whole Genome Alignments. And for each of the different divisions which have this data, so Ensembl bacteria does not have this data, we can have a look at what species have alignments available. We can click on Alignment List, and this shows us all of the species that have alignments and what types of alignments they have, similar to the other page. So we click on Arabidopsis. You can see we have a list of all the species here, and all of the different methods that we use to create these alignments. And you can also quickly look at examples. So let's have a look at Brassica oleracea. And you can see that we've come to a location page similar to what we saw on the Ensembl main site, with the options to view Synteny alignments, both text and image, and also region comparison. Okay, so I think that has comprehensively covered the different sequence-based comparative genomics resources in Ensembl. I hope you found this video useful. Please do contact us if you've got any further questions at helpdesk.ensembl.org, and otherwise, happy browsing.